Thanks everybody for coming. And so today I'm going to talk about how we are protecting our areas which support biodiversity. So as Ray said, I'm part of the Aquatic Pollution Prevention Partnership, or A3P. And so I'll be talking about the work that we're doing to protect environmentally sensitive areas in around Melbourne. So the key research questions that I would like to ask for this project are, what do we know about the risks to areas of biodiversity significance? So for example, Melbourne Water's sites of biodiversity significance, Ramsar sites and other environmentally sensitive areas. And what are the appropriate management responses to manage the key threats to these environmental values? And this project will address some of the regional performance objectives, and they're listed here in this slide. So the project initially focused on the Melbourne Water's sites of biodiversity significance, or SOBs, but these are areas that Melbourne Water manage or own, and they support key environmental values such as vegetation, birds, frogs, macroinvertebrates, fish and platypus. And there are about 40 SOB sites around Melbourne that Melbourne Water manage, and you can see them here in the blue. I don't know if you can see my pointer. So these are all the sites in blue here are the sites of biodiversity significance, and they range across the whole.
We can see your pointer, Sarah.
Oh, great, thank you. So they range across the whole of the Melbourne Water region, and they differ in size as well. So you get small sites like Yering-Back Swamp in Yarra Glen towards the northeast of the region, and then you've got the Western Treatment Plant in Werribee that's a very large site. So this is the approach that we took for this project. So as I mentioned earlier, there's over 40 sites that Melbourne Water manage, so we focused on the SOBs. And so we're not able to assess all of those sites in the first few years. So we needed to develop a strategy to prioritise which sites that we're going to look at. So I carried out a desktop review and looked at the risk of contamination at each of these sites. And so I'll talk about the desktop review a little bit later on. So once we'd identified the sites that we were going to sample, we needed to do a site visit. So I visited all the sites that we'd identified and met up with the Melbourne Water, Waterway and Land officers. So I just at this point want to thank all of those WLOs that really helped with this project for their time and also their knowledge around these sites. And also as well, staff from the local councils and also Parks Victoria who've been really instrumental in transferring their knowledge and sharing the knowledge with me about all the different sites that we're interested in. So we looked to find suitable sampling locations, looked at access, also looked to see if there's other inputs into the SOBs that could potentially be influencing the water quality. And so once we've done that, we then went back and collected and analysed sediment and water samples, and mainly focused on urban contaminants. And we also deployed passive samplers. So I'm not sure if everybody's familiar with passive samplers, but this photograph here on the bottom right is an example of a couple of our passive sampling devices. So they get deployed within a waterway and we put them in there for about 4 weeks. And so any contaminants that are present in the water will flow across the membranes, and the different membranes will attract different types of contaminants based on their chemical properties. And so after we've deployed these passive samplers, they're in situ for up to about 4 weeks. We retrieve them and then we can extract what's left on the membrane. So they're really useful for getting a sort of time integrated idea of what's passing through the waterways over the 4-week period. So we put the passive samplers in the SOB site, but also in the drains and creeks that flow into them in case we needed to track sources in future. So once we've got all the data, we then have another project meeting with the Melbourne Water team as well as RMIT, and we'd identify sites that needed further investigation if these did occur. And so for that, we'd use a weight of evidence approach, which I'll discuss a bit later in the talk. So the first thing we needed to do was to actually look at what land use was. So from work that we had completed earlier on in the partnership, we'd identified particular land uses that are associated with particular types of contaminants. So when we created these land use maps, you can straight away see that there's different land uses associated with around the different SOBs. So if we look at Old Joe's Creek Retarding Basin, you can see the SOB site here in blue, and the different colours represent different types of land uses. So the residential's in purple. So you can really see that around Old Joe's Creek retarding basin, there's a big residential influence, a little bit of green space, and then there's some industry further down the catchment. Whereas when we compare that to Cherry Lake, we can immediately see that it's a bigger site, but also there's different activities going on in the catchment. So you've got industry further up the catchment, you've got some green space and also some residential. So we can see straight away that there's going to be different contaminants present at the different areas based on their land use. And we know from previous work that industrial areas tend to be more at risk, waterways that are in around industrial areas tend to be more at risk from contaminants. So we could see that these two areas are likely to be influenced. So we looked at the land use, and we also, I also read the management plans for each of the SOB sites. And this will help me identify the different values that were present at each of the sites. And also if there's any previous threats that had been identified that could threaten the biodiversity of each of the sites. And that really helped me with developing the risk assessment matrix that we used in the first part of this project. So then we needed to create the risk assessment matrix. So these are the different criteria that we used. So like I said, we looked at the environmental values that were present at each of the sites, and we focused on the values that we considered to be more at risk to contaminants. So these were frogs, macroinvertebrates, birds and fish. And we also considered other threatened species that aren't listed in Healthy Waterway Strategy, but we did think were important. So things such as the swamp skinks. We also looked at previous reports to see if there's any likelihood of contaminant risk and to give us a better understanding of what could be present. And then, like I said earlier, we know that this land use and contaminants are really closely linked. So we listed all the contaminants present at potentially present at each of the sites based on the land use. And then with that data, I then looked at publicly available databases, relied on prior knowledge, and also looked at the literature to try and see if there's any impacts of these contaminants on the values of each of the sites. And that was really important because it really helped me develop the risk score for the risk assessment. So for an example, if we saw one of the land uses and we knew that particular contaminants were present, when I went and had a look at the toxicity data, if that contaminant isn't likely to be toxic to the values that were present at that site, then that site would get a lower risk score. So then after I'd completed my risk assessment, together as a project team, we looked at the ranking of the sites, and we'd worked out which sites we would focus on in the first year and then subsequent years after that. So the first year we chose sites that were geographically spread across the Melbourne water region, and also we focused on a range of different environmental values. So these are the sites that we looked at in the first year. So what we did was we looked at the northern region, the western region, and also the southeast region. But just to point out that the Western Treatment Plant came out as the highest ranked site, but we excluded this site at this stage because there was already an ongoing programme of work around this area. So we wanted to focus on sites where there wasn't as much information available so that we could expand the knowledge base for each of these SOB sites. So then in the second and third years, we expanded our sites. We went back to our risk assessment matrix and looked at some of the medium risk sites. We also wanted to validate our risk assessment. So we looked at the sites that we considered would be low risk, but contained really high values. Just to validate our risk assessment to see if these sites really were under low contamination risk. And then we also expanded our sites into non-SOB sites, but sites that support a wide range of biodiversity values. And then in the third year, we went back to look at some high value, low risk sites. We were hoping to sample these sites in year 2, but because of the COVID interruptions, we weren't able to sample at these sites. So we've sampled these sites now and also carried out some follow-up investigations from years 1 and 2. And then it came to our attention through recent reports that there was some key knowledge gaps at the Western Treatment Plant. So we're actually going to go and work on the Western Treatment Plant for the rest of this year and also next year to try and address those limitations in the knowledge. So I just want to talk you through some of the types of follow-up investigations that we are undergoing as part of this project. So we tend to use a range of tools within the A3P toolbox. So tools that we can track the source of contamination, but also we can determine whether the contamination is likely to have impacts on the biota. And this will help us to recommend appropriate management actions to reduce the contamination risk at these really important sites. So as discussed in last week's talks, we use a multiple lines of evidence approach. So we use a range of different tools, from chemical analysis of sediment and water through to deployment of passive samplers. And also we're looking at some biological approaches. And one example of that is to assess the toxicity of the sediment or water to a range of species that we have at RMIT. And they include invertebrates and also plants. So the first slide on the left, the first photograph on the left shows a picture of a toxicity test. So one of the sites that we looked at had levels of metals that were above trigger values. So we wanted to know whether these metals were likely to have an impact on our invertebrates. So we sampled a range of locations within that site, brought the sediment back to RMIT and carried out some tests to see if our invertebrates were impacted by the sediment. And we did find that there was some impact on growth. And when we compared the results for the toxicity test with the metals, we realized that metals were no longer an issue. So we're now following this up to find out what it is that's causing the issue. And so we're deploying passive samplers to try and pick up some soluble chemicals that could be present. Another type of approach we use is to do comprehensive sediment surveys to try and track the source of contaminants. And then finally, we'll also do annual monitoring of our high value sites. So this is more to use an early warning system to see if we start to see any degradation of these really high value, low risk sites, so we can start to implement management responses before hopefully any impacts will occur on the values. So to sum up, this project has contributed to meeting the Healthy Waterways Strategy performance objectives to protecting sites of high biodiversity in urban and rural areas. It's determined if pollutants are an issue at environmentally sensitive sites. And it's identified sources of contamination and recommended management actions to reduce contamination at these sites. So I'd just like to thank everybody on this slide for helping with this project, and thank you for listening.
Great, thanks very much, Sarah. That's excellent timing and some really important work there in terms of knowledge gaps around toxicants and these important sites of biodiversity significance.
Thanks, Reece.
So